You're probably aware from 701 in high school biology what proteins can do, but let me just remind you, what is it that proteins do? Well, they do a lot of things, and I'm just going to illuminate a few for you here. One of the things that they clearly do, right, is catalysis. We'll talk a lot about this in future lectures. They enhance reactions or reaction rates because they often function as enzymes. What else do they do? They provide structural support. At the macroscopic level, for example, by proteins like collagen. It makes up your bones and joints and tendons, and also your jello. Right? And at the microscopic level, tubulin and actin. It make up the cytoskeleton inside individual cells. Transport. Hemoglobin, myoglobin, the molecules that are responsible for transporting oxygen in the blood and in the muscle that you'll learn about shortly if you don't already. There's another molecule called ferritin. Fe gives you a hint. It's an iron transporter. You need a lot of iron in your body, and ferritin is what helps store it and transport it. Movement. The fact that you can write notes happens because, of course, there's muscular contraction driven in large part by myosin, okay? A motor protein. Signaling. Signal transduction. Things that control, for example, heart rate and blood pressure, which, as you will learn in one of the future lectures, is controlled by receptor proteins. G proteins and kinases, all of which we will discuss shortly. Vision, controlled by the molecule in your eye called rhodopsin. That's what lets you see me so that you can then take the notes in your notebook here. Okay, ion pumps and channels. All of these things are built from the same set of 20 amino acids. Pretty incredible. But it means that if we want to understand what proteins do, we want to isolate them and try to figure out how they work. We want to study their function and make biochemical measurements and biophysical measurements, solve their structure. We're going to have to purify them. And so to do this, we have to talk about the steps that we're going to use to purify the proteins that are responsible for doing the biological activities that we're interested in. So now we're going to move on and talk about purification steps. Where do we start? Well, the first place we start is we got to break open the cells. Now, there's some exceptions to this, right? Some of the first proteins to be studied, nobody had to break open any cells. That's because they just studied proteins in your blood, right? We could just take blood from you, spin the cells down, take what's left that's not clotted called plasma. We'll talk about that shortly. But if we want to do this, we have to find a way to break open the cells. And there are a bunch of techniques we can use. We can grind the cells up, right? We can either grind them up by themselves, we can throw in some sand, we can do anything we want to grind the cells up. We can freeze and thaw them. Freeze, thaw, freeze, thaw, freeze, thaw. You break the membranes. That's one way to do it. You can use a detergent, something that disrupts the cell membrane. You can use sound waves to shear the protein apart. My favorite, and the one that's probably the best of all of these, is rapid pressure changes. I should have written the word rapid. Rapid pressure changes. Okay, an example of this is something called a French press. I know. A French press is that thing you put the coffee in the morning and you pour the hot water. No, this is a different French press. Why does rapid pressure work? Well, imagine that I take some cells and I put them in a high pressure bomb and I turn the pressure up to 500 atmospheres. Okay? What happens? Well, the cells equilibrate, right? The pressure outside is very high initially, but the cells rapidly equilibrate. And now the inside of the cell is at 500 atmospheres, and the outside of the cell is at 500 atmospheres. Now what do? I release the pressure outside. Now what happens? Suddenly inside the cell is 500 atmospheres, outside the cell it's 1 atmosphere. Kaboom! The cells explode. Works really well. Very, very gentle. So now I've got some cell goo at the end of this, right? And now I want to figure out what I'm going to do with this. So I got to go one step further after I've broken the cells open, and I got to somehow separate this into the next steps. So to do that, the next step I'm going to do is I'm going to separate them by centrifugation. Okay? So I'm going to take my cell goo, and I'm going to spin it at, say, low speed, which for this class, let's just say 500 times gravity, okay, for 10 minutes. And I'm going to end up then, in my tube, I'm going to end up with a pellet, and I'm going to end up here with some supernatant. And when I do this, in fact, the pellet at this speed contains nuclei. So at this point, I'm going to take the supernatant, and I'm going to spin it again. This time, I'm going to spin it at medium speed. And medium speed here is about 10,000 times g. And I'm going to spin this for about 20 minutes. And I'm going to end up again with a supernatant and a pellet. And the pellet that I end up with this time contains mitochondria. Now, at any point in time, I can stop, right? I can say, oh, I'm really interested in what's going on in the nuclei, and I can work with them. Or I can say, oh, I'm interested in the mitochondria. But if not, I still have a bunch of stuff here. So I could take the supernatant again, and I could spin it again, this time at high speed. And by high speed, I mean 100,000 times the force of gravity for, say, 2 hours. And now I'm going to end up with a pellet and a supernatant. And my pellet, when I do this, will be primarily microsomes. And microsomes are nothing more than ribosomes and membranes. Basically rough ER. And I'll end up with a supernatant here. And this supernatant that we're left with here, we're going to call cytosol. That's the sap inside cells. Okay? So the supernatant at this point equals cytosol. Okay? Now, let's just say my protein is sitting here in the cytosol. So now I need to do the next step in order to work to get that out. So to do this, now the next thing I have to do is I'm going to make use, I guess I should write this over here. How am I going to get my protein out of this cytosol? Well, I'm going to now use differences in solubility, charge, size, or affinity. One thing I can use is differences in solubility. And that technique is what we call salting out. I'm going to make use of the fact that in certain salts, certain ionic conditions, I can make proteins more or less soluble. So the idea here is that as I increase the ionic strength of my solution, I can decrease the solubility. But in order for this to work, I have to have a salt that's very soluble. And there's a very special salt that biologists use, biochemists use, and that's the salt ammonium sulfate, NH42SO4, ammonium sulfate. It's a special salt because it's really, really soluble. That means I can get the concentration of it way high up, and it's pretty gentle on proteins. You can use sodium chloride, which is what molecular biologists who think they're biochemists use, but it's a much harsher salt. If you know about the Hofmeister series, you'll recognize why. Let's consider, as an example, two proteins in serum. How do I get serum? Easy. Just to remind you, I take blood, I don't let it clot, I spin it, I get cells down here, and I get what's called plasma here. That's what I'm interested in, not serum. Apologies. Plasma. If I let it clot, I have serum. Plasma is the unclotted part of your blood. Okay? Now, there are two proteins that are sitting around in plasma, and I want to purify them. Okay? One of those proteins is called fibrinogen. We'll talk about this in a future lecture. This is a protein that's involved in clotting. And the other thing I'm interested in is albumin. This is a protein that does a lot of things in your blood. Now, the interesting thing is that fibrinogen will precipitate out at 0.8 molar ammonium sulfate. Albumin will only precipitate at greater than or equal to 2.4 molar ammonium sulfate. So now I have a slick trick. All I have to do now, right, so in order to do this, all I have to do is take plasma and add to it ammonium sulfate so that its final concentration is anything greater than 0.8 molar. Let's go with 2 molar. For example, I could have used 1 molar. I chose to use 2. What happens at 2 molar? The fibrinogen precipitates. The albumin does not. Okay? And so then I can centrifuge. And I'm going to end up then with a pellet that's rich in fibrinogen. And I'm going to end up with a supernatant that still has albumin in it. It may have other things, but it still has albumin in it. Thank you, ladies. Okay? Then what do? Then I take the supernatant and I add more ammonium sulfate. Let's pick a number. I don't know, 2.8 molar. Could I have made this 3 molar? Yes. Could I have made it 4 molar? Yes. Could I have made it 2.6 molar? Yes. Anything greater than 2.4. Okay? And when I do this now, if I centrifuge, I will end up with a pellet that hopefully is primarily fibrinogen. Sorry, albumin, right? And now I'll end up with a supernatant that has some other stuff in it. Why didn't I just make the concentration 2.8 molar at the beginning? What would have ended up if I'd gone from 0 to 2.8 molar? What would have been in this pellet? Both, of course. So I can differentially, by stepping up the ammonium sulfate concentration, I can preferentially now separate one protein from another from another. Okay, so now I got this albumin pellet. I take the pellet and I want to work with it, but I got a problem, don't I? The problem is that my protein is sitting now in this salt-rich precipitate. So what do I have to do to get rid of all that salt? Well, it's precipitated. What do I have to do to get it to go in solution first? Dilute it, right? So now I got to get that ammonium sulfate less than 2.4 molar. Okay, so I'm going to take my pellet, I'm going to add a lot of buffer, I'm going to dilute that salt, suddenly that pellet dissolves in solution, right? And now I have to do dialysis, because I want to get rid of that dialysis now to remove the ammonium sulfate. Now you guys, I'm sure, I hope, learned about dialysis like in high school. But if not, I'll remind you what it is. I'm going to take our protein, our albumin, which I'll make purple. Okay, here's our albumin, purple. Okay, and now I've got a bunch of ammonium sulfate. which I'll make, let's pick a different color. Let's make it green. Let's make it blue. You know those nerdy kids, right, with the pens with like the 5 different colors, right? You know those? Can you guess I was one of them? Yeah. Okay. So now I got all this ammonium sulfate sitting in this dialysis bag, and I put this in. I'm trying to purify a protein. What do you think I should put it in, water or a buffer? Who says put it in water? Who says put it in a buffer? Of course, that's why we learned about buffers. So I put this thing in a buffer, and I wait, and as you well know, what's going to happen to my dialysis bag? The proteins are stuck inside that bag. That bag has tiny holes in it, holes that are small enough that it allows my ammonium sulfate to diffuse out, but it does not allow the protein to get out. Okay? So again, this is a type of separation based on size, but it's not very good for separating one protein from another. It's pretty good for separating small molecules from your protein. Okay, and again, dialysis, what we're really talking about here is a semi-permeable membrane. This technique, dialysis to remove the salt, works pretty well for removing salt from proteins. How do I separate the proteins from each other? Well, there's some ways that I can do that. I can do it by can I use size to separate the proteins from each other? Remember, I showed you, here's a protein, right, that's 104 amino acids, and here's a protein that's 34,000. So clearly, they're going to be very different in size. Can I use a technique to separate these proteins based on size? Well, I can. And that technique, based on size, is called size exclusion chromatography. Also called gel filtration chromatography. Okay. Now, gel filtration, the idea is I'm going to use a column that I'm going to fill with porous beads. Porous beads. And these beads can be a variety of different things, but they're usually a polymer of some kind of dextran, which is a sugar molecule, or agarose, or polyacrylamide. So what I'm going to end up with then is a column that looks like this. Now these beads are tiny. These beads are about 0.1 millimeter in size. And they have, so I draw them, they look like this. Here's a bead, here's a bead, here's a bead, here's a bead. Another one, there's another one. Okay, and these beads have tiny little pores that run through them. They're not necessarily the straightest pores, but they have all these pores that run through them. Here's the bottom of the column. Okay? Now, if I put in 2 sizes of molecules, I'm going to put in big purple molecules at the top, okay? And I'm going to put in pretty tiny orange molecules. Okay? Both are proteins. The orange and the purple are both proteins. What happens is the purple molecules can't fit into the beads, so they have to sort of work their way around in between the beads to come out. Time. Okay? And what I find is that that the purple stuff that works its way around the beads comes out first. In contrast, these orange molecules, they can go through the beads, but they take a long time to work their way through the pores of the beads. And so I find that the orange molecules come out later. Okay, and that's what's shown in this slide here. You can see big yellow molecules, intermediate green ones, really small red ones. The big ones, the yellow ones, come out first. The green ones come out somewhere in the middle. And the red guys that can really get lost inside the pores of the beads come out the last. Okay? Now, to make this technique work well, should I use and these are examples of columns, okay? 2 real columns. If I want to separate something by gel filtration, should I have a long, thin column or a short, fat column? Who says short, fat column? Who says long, thin column? Think about it. You want them to separate over time. So the longer the distance that they have, the more they will separate. Yes?
How would you know when the protein actually is coming out?
You don't. We'll get to that. You're just if you can't see it, you're just collecting fractions and you're going to figure out where the protein was. Okay? So for this, I want a long, thin column, not a short, fat column. Okay. Should I put a lot of sample on or just a little bit? Should I put half my column volume of sample or maybe 1/30th, 1/40th? Who says a whole lot? Who says a little? Yeah, the smaller amount gives it better separation. So this is separation based on size.